Now we're going to look at movements through cell membranes. First, we're going to start with diffusion and osmosis that don't require an input of energy. Diffusion is movement of molecules from an area of high concentration to an area of low concentration. So if we're imagining molecules, and a lot of them here, not so many of them here, they're moving from high to low. This is down a concentration gradient. Diffusion occurs until equilibrium is reached. For example, when a crystal of dye is placed in water, the dye and water molecules move about until equilibrium occurs. In a cell, for example, oxygen and carbon dioxide move by diffusion. Oxygen and carbon dioxide were small molecules that were able to easily pass through the plasma membrane. In a solution, we have a solute and a solvent. That solute, shown here, is a solid that becomes dissolved within a liquid or the solvent. We talked earlier about how water is the universal solvent since a lot of things dissolve in it. Once the solute and solvent are evenly distributed, their molecules still continue to move about, but there's no net movement of either one in any direction. So here's your crystal of dye. It's a high concentration of the dye, and it's going to move to an area of low concentration. So these molecules move about until they're about evenly distributed. Again, there's no input of energy here. With facilitated transport or facilitated diffusion, this helps explain the passage of molecules such as glucose or amino acids that don't easily cross the plasma membrane. Neither molecule is lipid soluble. If we remember, those hydrophobic tails of phospholipids form the interior of the plasma membrane. Like diffusion, in facilitated transport, ATP or energy is not required because molecules are still transported down their concentration gradient. This is how that looks. With a carrier protein that spans the plasma membrane, this allows for the passage of small molecules that are not lipid soluble. Molecules still follow their concentration gradient and energy is not required. So the solute that is outside of the cell here, here's a higher concentration of the solute here, low concentration here. This carrier protein can change conformation or shape to help transport it. Osmosis is a special type of diffusion. It involves water molecules across a differentially permeable membrane. Another way to say that is a semi-permeable membrane or selectively permeable membrane. All that means is that some molecules can easily cross and others can't. Similarly, diffusion is always going to occur from an area of higher to an area of lower concentration. Osmotic pressure is the pressure that develops in a system due to osmosis. The greater the possible osmotic pressure, the more likely it is that water will diffuse in that direction. Here we have a differentially permeable membrane. Water molecules can easily cross, but the solute molecules cannot. Here, if we have a greater concentration of water, more molecules, here is 5% representing solute concentration. So water molecules are going to try to equalize across that membrane. In osmosis, we're comparing solutions to see the likelihood of water molecules moving one way or another. Here, we're going to compare the interior of a cell to the exterior of a cell, as though we place a cell within a solution. If that solution is isotonic, iso means same. The solute concentration is equal inside and outside the cell. In a hypotonic solution, hypo means lower. If you think of hypothermia as in a lower temperature, a hypotonic solution has a lower solute concentration than the inside of a cell. Hypertonic is just the opposite. Hyper means higher. Think hyperactive or overly active. So a hypertonic solution has a higher solute concentration than the inside of the cell. If we're looking at an animal cell, such as a red blood cell, a 0.9% sodium chloride solution is isotonic. So that means there's an equal solute concentration in and out of the cell. If you place a red blood cell in a 0.9% sodium chloride solution, there is no net gain or loss of water. This is good if you're wanting the cell to maintain its shape and function. In a hypotonic solution, there is a lower concentration, so water is going to move into the cell. If so much water moves in that the cell can't handle it, then that plasma membrane is going to burst. This is lysis of the cell. In a hypertonic solution, we have the opposite occur. So hypertonic, there is more solute, and so as water tries to equalize, water leaves the cell and causes the cell to shrivel or crenate. In a plant cell, this is similar, but we have a cell wall here as well as a central vacuole and these chloroplasts that can help tell us what's going on. In an isotonic solution, there's no net movement of water, right? We have an equal amount of solute particles on either side of the cell. And so we can see that there's a fairly equal distribution of chloroplasts within these plant cells. In a hypotonic solution, the cell is going to gain water as it tries to equalize. In this case, the central vacuole fills with water. We saw earlier how that central vacuole is for storage and support in a plant cell. Here, it's able to store that excess water. As it expands, it pushes chloroplasts to the outside of the cell, shown here. You have turgor pressure, which also develops. In a hypertonic solution, this is just the opposite. The central vacuole loses water. The cytoplasm shrinks, which is plasmolysis, and the chloroplasts gather towards the center of the cell, seen here. If we're comparing animal and plant cells, we have the same basic thing that happens during each of these solutions. Isotonic, no net movement. Hypotonic, you have an intake of water. In a hypertonic solution, water leaves the cell. So with diffusion and osmosis, we are moving down a concentration gradient. There's times when you want to move up a concentration gradient. So this is where active transport comes into play. Molecules or ions combine with carrier proteins, often called pumps, and molecules are moving against the concentration gradient from an area of lower concentration to an area of higher concentration. This is similar to swimming upstream that requires energy. Or if you think of trying to pack people into a room, that requires energy. This can be active transport entering or leaving the cell. In either way, you're going to accumulate a concentration gradient on one side of the plasma membrane. The key here with active transport is that energy and carrier proteins are required. That energy is usually in the form of ATP. The sodium-potassium pump is an example of active transport. In this, the carrier has a shape that allows it to take up 3 sodium ions, and ATP is that energy that is used, and it's split. And so ADP is an adenosine diphosphate. That triphosphate is here, that third one. This changes the shape of the carrier protein, and so sodium ions can be released, and 2 potassium ions can be taken up, and those are pushed to the other side, and a concentration gradient can be established here. Other ways to move substances in and out of cells, we have exocytosis. In exocytosis, substances exit the cell, but here we have the use of vesicles. Vesicles were those tiny membranous sacs that transport substances. Here, the vesicle fuses with the plasma membrane as secretion occurs. The vesicle membrane becomes part of the plasma membrane. Cells of particular organs are specialized to produce and export molecules. For example, our pancreatic cells release insulin or enzymes. The anterior pituitary cells release growth hormone. So if we're envisioning exocytosis, we have this phospholipid bilayer of a secretory vesicle, little membranous sac. Here it fuses with the plasma membrane, becomes a part of it, and that substance can then be released outside the cell. So exocytosis, exiting the cell. With endocytosis, cells take in substances by vesicle formation. This is just the opposite of exocytosis. Part of the plasma membrane invaginates to envelop the substance. The membrane then pinches off to form an intracellular vesicle. We have two types of endocytosis: phagocytosis or pinocytosis. In phagocytosis, you have a large particulate matter such as food molecules or viruses or whole cells that are entering. An amoeba or macrophages are examples of this, where there's eating or breaking down of matter. In pinocytosis, pino means to drink. Liquids and small particles that are dissolved in liquids are taken into the cell. Certain blood cells or plant root cells are examples of this. So with phagocytosis, this is cell eating. The cell takes in solids. So if you have paramecium and the pseudopod of an amoeba, you have a vacuole forming there. Another example is white blood cells use phagocytosis to destroy bacteria. Pinocytosis is cell drinking. So whether you have a liquid or something dissolved within a liquid, then you have a vesicle that forms. And again, with endocytosis, things are being taken into the cell.